Good afternoon, everyone. My name is Raina Ghazi, and I'm the Teacher Development Manager for Macmillan Education. Thank you so much for joining our webinar this afternoon in the UK with Neva Miles, who's going to be focusing on human and social biology. If you have any questions, feel free to write them in the chat box, and we'll do our best to answer your questions. I will pass it over to Neva now. Thank you.
Thank you, Raina. Good afternoon from Great Britain, but I understand it's good morning where you are. So good morning, everyone. Thank you for joining us today. I've had the privilege of rewriting the Human and Social Biology book for the CSEC exams, and I've based it obviously on Phil Gadd's very popular 6th edition, and we have just maintained the academic integrity of the original edition while matching the text to the new syllabus and of course including features to support the school-based assessment and other features requested by teachers to make the content relevant to the real world and to make use of everyday equipment where possible. Next week we actually have a webinar to describe the features in the new edition in more detail, so I won't go into those too much, but there are a few examples in this presentation to give you a flavour of it. So who studies human and social biology? Well, I think there are probably two main groups. The first group are the learners who struggle with physics, chemistry, and maybe biology. They may be studying PE. They may not be succeeding in maths, of course. They may be struggling with reading and writing even. There may be many reasons why they struggle with science, and there's no reason why aspects of the HSB syllabus could be considered easier than other sciences. So we need strategies to overcome the more difficult concepts for these students. And then the second group of learners are those who are studying other sciences and in some instances are encouraged to take HSB as an extra subject. And for them, independent learning is the key with minimal support from the teacher. So some of our strategies could help those sorts of learners too. One of the strategies that a lot of people have been using these days is something called blended learning. And the left-hand diagram there shows that the basic view of what people understand by blended learning. It includes face-to-face learning in the classroom and online learning, obviously using the internet. And overall, you get a blended learning approach. But I actually quite like the other diagram, which shows far more what actually goes on in most classrooms. Not only are the students having challenges in this subject, but also the teachers. And teachers have actually indicated to us that there's a lot of theory to be covered. I think you'll appreciate that from the syllabus. Time is limited, and there's a lack of space and equipment in many schools, and that's a real problem. And getting beyond knowledge to developing understanding has been something that some teachers actually feel very worried about. We can't solve all those problems in 1 hour, but I hope there are some ideas here for everyone. The experienced teachers among you and those who'll be starting the syllabus perhaps in September. So let's start by looking at these ideas behind blended learning, and then we can move on to some topics that learners find difficult. This slide shows you the sorts of strategies that you're probably already using to some extent in your classroom. And blended learning actually weaves methods, various methods, into a cohesive whole. It combines classroom teaching and digital learning. Lessons could actually begin in the classroom and continue online, or you could flip that around and suggest that they start looking at something online and then come back to the classroom to develop it further. It features obviously multiple teaching methods. The digital learning is used to provide additional content when paired with traditional instruction. You probably appreciate that nowadays teachers are often expected to do more with less. So in a blended approach, traditional instruction is used to introduce or reinforce concepts, focus on hands-on skills practice, and provide opportunities for collaborative work. Along with your textbook, try supplementing your lessons with free resources from the internet, online discussion groups, your homegrown teaching resources, and test preparation materials. Some topics, of course, are actually better shown with a video, blood flowing through blood vessels, for example, or experiments when laboratory is not available. You could also try a mobile app or an interactive game if one fits your courses. So, blended learning provides teachers with flexibility, frees you from the race against the clock. It can also meet the needs of anxious students. Online discussions with their peers, time to consider a question or articulate it, to share their responses, and it gives them an equal voice, and there's no pressure. It allows you to incorporate methods suitable for all the learning styles, the 4 learning styles, of course, being visual, auditory, reading and writing, or kinesthetic, more active activities. And here are some of the sorts of things that you're probably using already in the classroom. Students using models, students taking their own photographs, perhaps, of experiments maybe, or work outside when they're doing fieldwork and so on. Students doing presentations. I always think it's a good idea. For example, if you're doing something on vitamins, have a different child, student at this level, actually giving the presentation on that particular topic. Maybe putting a big badge on the front of them, I'm vitamin D, and then when learners try to remember what's going on, they can relate that vitamin to that person and what they actually said on the day. Of course, they're using computers, they're using laptops in some cases. Many of them may well be using their mobile phones, which can be a bit more tricky if you're trying to show several videos. I've included one picture of children looking really animated when they're doing probably a chemistry experiment from that. But actually, I couldn't tell what they were actually trying to show. Is it just showing a change in colour? And they're looking quite stunned by it. But are they actually learning? Do they understand what's actually going on during that experiment? And this is one of our problems, of course, for learners who are less able. It's not the what, but the why that's important. And the usual practical lessons, of course. And then if you want to change the face of the educator, learning at home has become quite a new thing, I suppose, with the pandemic. And use parents and grandparents where possible, or carers of children. Suggest that they try things with them that they might not do in school, but discuss with them some of the things. So, for example, they might think photosynthesis isn't relevant to human biology, but actually it's a very important part of our lives, and maybe doing something in a garden or in a park or some other facility might be a good idea. When you're going on trips, of course, using another person to be the expert for the day can also engage the students, and they often ask questions there much more readily, perhaps, than from their own teacher. Bringing in visitors, for example, they have to do CPR. So in this case, we definitely have changed the face of the educator. We've got a model, but you've also got an expert then to explain how to use it properly and how to do these things where we might not be the experts ourselves. I quite like the idea of using the human body as the resource. If you think about students going into exams, they're actually taking the physical resource in with them, which isn't the case for any other subject. And if they've been in the habit of labelling parts of their body, as these youngsters are here, it can be a good memory trigger when they get into the exam. I mean, one of the problems with exams, well, with learning, I suppose, is not getting the material into your head. Very often it's getting it back out again. And so by having triggers, it can be a really good thing to do. And for example, if they're learning the brain, actually put their hand on the head in the region where they're thinking or where the sensory organs are linked to and so on. And doing that. And in the exam, maybe they can think, well, put their hand on the head. No one's going to tell them they're cheating. And they can actually perhaps trigger a bit of a memory of what they were doing. We've got 2 younger children here sitting together. doing this sort of pairing and sharing idea, they're teaching each other. And there are a lot of activities I'm sure that you do already, using flashcards or matching functions to organs in the body and so on. And talking together and sharing can actually enhance their learning. And they remember better sometimes when they work together as a pair, or maybe a small group, of course. I've got one activity. I was spoiled for choice here to actually give you an example of how a simple experiment could be seen at home. This is the biuret test for a protein, and the activity in the textbook is there. They've got all the prompts that tell them how to do the actual test if you've got the facilities, but they could also go online and watch the video with the teacher actually doing it for them, so they can see what happens. And of course, we got the results there actually in the books. We tried to do that where possible to show the results of experiments, and that's quite useful. But seeing somebody actually doing it, and of course you could actually make your own videos. I have no idea how you're managing lockdown at the moment, but maybe many of you are already producing videos of experiments and so on, and others might like to share those or do their own and get the ideas from their colleagues. One of the ways to make the subject actually relevant, where lots of children, they do a subject and think, why am I doing this? What's the use of it? How am I going to use it in the real world? And in the book, we've actually got lots of these real world situations. I've just given you an idea of two of them. One particularly aimed at PE students who might not have to learn all about strains and so on and fractures for this particular subject, but actually they'll find interesting because it links in not only with their human biology, but also with their PE. And by offering a few ideas about careers, and here we've got careers in muscles and bones, physiotherapists and chiropractors and so on, by offering those, it prompts some students to think, well, actually, maybe I could do something when I leave school in this sort of area. I think many human and social biology students not doing another science might feel, oh, well, perhaps I should actually try another science when I've got the opportunity, so that I've actually got the qualifications perhaps for moving on. So it's more aspirational for some of them. But those students who are already doing science and maybe doing human and social biology separately, they will get ideas too about the sorts of careers that they might be able to undertake. And here we've got some of the things that students actually find difficult. And we really need to know how we could tackle these problems when, in fact, we're short of time. We're worried about understanding them as well as just knowing all the knowledge and so on that the textbook shows you and the syllabus expects. So these are the ones that teachers have told us that they often find that students find really hard to learn. Circulation is one of them. I'll come back to that later. Excretion in the kidneys always seems to be a tricky one. Enzymes are difficult largely because of being short of lab time. And then other topics that people say that their students have difficulty with is genetics and inheritance, and particularly mitosis and meiosis. So we'll have a little look at that later on. And of course, the book has a huge amount on diseases. And I think that's something that you have to rationalize. So I've tried to show how if you can pick out the key points, it might help to reduce the amount of learning. So let's start with the kidney. And here, what we're doing is starting with the familiar. If it's a new topic, obviously they'll have had some idea of some of the words associated with the kidney from earlier stages in school, and maybe even in primary school, where they learn a little bit about the human body. So we need to have some understanding of what they already know. So let's start the lesson and start this topic with, okay, let's sit down and write down everything we can think of associated already with the kidney. Limit the time. I've suggested 1 minute. I would think 1 or 2 maybe. And students write down as much as they can, whether it's right or wrong, it doesn't matter. And then spend a short amount of time perhaps discussing their suggestions. And then lead that into, well, hopefully they've sort of said something about waste, but discuss why we would need to remove waste materials. Where are they? How can we separate them from the blood? How do we separate things? And I think that's probably the place to start. How do we actually separate things? So I've suggested that you can make a model nephron, and the book gives the not only the description of it, but also the items that you're going to need to actually make a model nephron. And the suggestion is, if you've got it, that you use a funnel with a filter paper inside, but if not, a sieve, or even, I call it a colander, you know, when you're putting all your vegetables through to drain. And that's useful with a bowl underneath, so they can do that at home. And the idea is that you get a jar, you fill it with a few beans, lentils, beads, marbles, anything like that, large things and small things. Add a small quantity of glucose, any other sugar would do, and a small amount of salt. They should realise straight away that, of course, the salt and the sugar are not going to be there as crystals at the end of it. They're going to dissolve, so they're going to be in the water. Sometimes when I've done this, I've added a couple of coffee grains as well. So look, by the end of it, it actually does look a little bit like urine, but that's just optional. And then, of course, if you set up your filter, if you've got a tube, attach the tube to the funnel and then put the end in the bowl. Pour the contents of your jar and what stays inside the filter. What goes through? Where does it go? So now we've got our model, we've poured our materials into it, and hopefully by discussing the actual model, students will realise that actually it's the large things that stay in the filter. Everything else goes through. And of course, where does it end up? It ends up in the bowl. Now, would that be a good idea if you're actually filtering the blood and everything you filter goes straight out and straight down into the bladder and out of the body? We can't afford to lose everything. We can't afford to lose all the water. We can't afford to lose useful things like glucose and mineral salts, but we want to get rid of the urea. So all the filtrate in the model goes into the bowl. So what's the kidney going to do? And how is it different from the model? Okay, well, somehow it's got to get the useful things back. At this stage, we discuss how the model is different from real tissue. So if we're using a rubber tubing, obviously that's impermeable. But actually, what's the kidney made of? Well, it's made of cells. So if there's a tube in the kidney, it must be made of cells. I would suggest that you, alongside that, you then, let's go. Right, I've gone a little bit further on just to introduce this diagram to you. I would get them to draw a very large diagram, probably on an A4 sheet, as big as you can, of what is called the kidney tubule. And at this stage, you can say, right, okay, look, this is what it looks like, and compare it with our model. So what you've got is something like a funnel at the beginning where the filtering goes on, and then you've got a tube. But actually the tube's not a rubber tube, and it's got two twisty bits in it and a loop. So when we've got a picture, we can link the model to that actual nephron. And then we can suggest, well, okay, we haven't just got one filter in a kidney, we've got thousands of them. So there's thousands of small filters. And what must this nephron or this kidney tubule be able to do? And we've discussed the fact that if everything is lost, well, we'd have to keep on eating and drinking all the time to replace it. So if useful materials are filtered, they must be recaptured. And the tubes leading from the filter region have got to actually do this. So if we've got our large diagram of a nephron now without labels, and this could be on the board, of course, we have to think, well, where do these functions take place? So first of all, where does the filtering take place? And hopefully they'll spot that actually that cup-like shape at the top of the tubule is where you'll get the filtering, because that's similar to our model. And then we have to think next, okay, well, the next thing that happens is we've got to think, where are things going to be reabsorbed? Sorry. Once you've actually thought about your nephron and what's going on there, if you've got a large picture on the board, you can put sticky labels on it. You can say, okay, we've got filtering going on in that first part of the tubule. And then you can say, well, all the rest of the stuff is going through the rest of the tube. So where is the sugar taken back? And I would suggest at this stage that you might even put a piece of flexible, perhaps garden netting of some sort around your rubber tubing to represent the capillaries, and say, well, it's got to get from the tube and it's got to get into the blood. So we need to have the two closely related. But at this stage, simply show a nephron and say, okay, where is the sugar taken back? And you can tell them it's the first twisty bit. Notice no special names yet. So we have a big label on, a big sticky label or a big label on their diagrams to say, this is where glucose is reabsorbed and other useful things. And then we'll say, okay, we've got to get some water back. Otherwise, if it all goes out of our body, we're going to get dehydrated. So which part of this tube is actually going to take the water back? Oh, it's this loop, right. So we'll label the loop, water reabsorbed. And then you say, well, okay, we've still got a few things that need taking back, and the water's got to be adjusted. So that second twisted tube is largely where the salt is balanced, and some salt is taken back into the blood. But if we've got too much, then perhaps it's going to carry on and go out with the urine. And finally, we've got the collecting duct at the end, and we say, okay, now we've got a tube that carries the urine out, a very small amount because it's a small tube, but this joins up with lots of other tubes to form the ureter at the opening of the kidney, and that ends up in the bladder. Now, once we got to that stage, we can say, okay, now we understand what the kidney actually has to do. It has to filter first of all, and then of course it has to reabsorb. And so far, we haven't used any of the complicated words. What we've tried to do is say, this is how a kidney works. So in a way, what I've tried to suggest is that we develop the understanding before we throw huge amounts of knowledge at them. So how are these filters now arranged in the kidney? This is the time probably to look at the diagrams of the urinary system in the book and look at the structure of the kidney itself. And they probably will see that in the cross-section of a kidney that you've got all those little lines. Well, that's sort of where all those tubules are arranged. And you can point out that the loops are in one region and the filters are in another region. They need to understand, of course, that the blood is carried to the kidney in the blood vessels, so that the renal artery is bringing the blood. Now, you've probably done that already in circulation. And then a reminder of what passes through the filter, what stays in the blood. And the key thing now is that they understand that big things stay in the blood and are not filtered. What are those big things? Well, they're the blood cells and the large blood proteins. Where does the glucose return to the blood? We've sort of discussed that already, and we've labelled our first twisted region with where glucose is reabsorbed. And then if you add those things to your labels, you'll find that they start to your diagram, I beg your pardon, they'll start to understand that this is quite a straightforward process. We've actually got just filtering and then reabsorption. And there's our diagram. And what I've suggested for a homework, or even you can do it in class, in groups or whatever, is that once you've got what's going on in the kidney tubule, in the nephron, then you start to add the clever names. So what do we call the filter at the start? Well, that's the Bowman's capsule. And inside that are the blood vessels, which allow the materials to actually pass from them into the capsule. Why does it pass? Because we increase the pressure. And so you just try to give them the idea then that perhaps the outer blood vessel is slightly narrower than the one going in, and that creates some extra pressure and forces materials through. So there's a bit of help in the filtering process there. It doesn't happen just by pouring materials as we did in the model. And all the time you need to get them to think, well, in what way is the model actually different from the real nephron? And we've discussed this already. Where is the salt reabsorbed? Put your sticky label on. What's left at the end of the tube? And they can make a list of the things that are left there. And where does it go? And I think I've covered that already. So once they've grasped how a kidney actually works, and you've completely added all the simple labels to the diagram, then this is the time to bring in all the other resources and record the new vocabulary. And if each learner has their own very large diagram, as we suggested, I think they'll find it easier than the smaller diagrams that they see. You know, if you read a book, you know that the small writing looks more difficult than if it's written in big print. And the same when you're learning music. If you see large staves with large notes on, it looks a lot easier than having a lot of staves on one page and quite small notes. So the same applies here. As the larger the diagrams, it often helps them to relate things to that topic. I've suggested that for a homework exercise, they could look for an online lesson on the kidneys. And I actually found one was called The Kidneys Part 1 to 3. Now, you may find it quite easily, but Part 2 is a clear description of filtering and reabsorption. There are a lot of different YouTube videos, and I think you need to look for the links. We don't include the links in the book because often things disappear or the links change, and that can be very frustrating. So what's important, I think, is that you check that you've got a suitable video, and this is just one suggestion. But this particular video had very clear animations. Part 1 actually deals with more detail of kidney function, and it even involves the ADH control of the kidney, which of course is a much more difficult part for them to learn. So I would suggest they don't look at Part 1 before they look at Part 2. So on the left, you can just see what we've been saying, really. We've developed the understanding before adding the detailed knowledge. We've used online lessons and images to allow learners to work at their own pace, maybe. And we can recommend selected videos. Right, now the next topic that people found tricky when they were teaching it was cell division. And I think, again, one of the problems here is that cell division and those sorts of topics have a lot of new words, and they get in the way of actually the understanding. And for some learners, it can be very abstract. So I think this is a classical one, where actually models, again, are very, very useful. And even if students can't make their own models, you could use them in the classroom. What they need to understand is that when a cell divides in the body, the nucleus has got to be identical to all the other cells. And of course, when eggs and sperm are made, they've got to contain half the DNA. And I've suggested for the activities that they actually either produce diagrams or posters, or you produce them, and they're good resources. But actually making the models can be really useful. I can remember my granddaughter actually having to make a model cell, and the class came up with all sorts of ways of doing things, and she actually made a cake. And it was quite stunning, to be honest. She had all the cell structures on that cake. But what was interesting, of course, when she took it in, it created interest, and the class were able to eat the cake, and it was quite memorable. So I imagine they'll remember far more than just from a textbook diagram by eating cake of a cell. I've suggested a pipe cleaner, so I'm sure lots of you have done this already. But an additional thing that I thought was actually very helpful when I was teaching was to create a spindle. And it was very easy to do because I happen to have copper wire handy and cork boards. And I just curved the thing up into a dome shape, flattened out the ends a little bit to push it into the cork board, and put several of these to create my own spindle. And then if you put pipe cleaners at one end, and let's say you have 2 pairs, so you have 2 long ones and 2 short ones perhaps, you could then put single ones. And I think this is where some students get confused, that we always show chromosomes on karyotypes as being already replicated. And actually, they replicate just before cell division. So what you need to start with are single pipe cleaners, and then say, okay, well, what's going to happen to these chromosomes? Well, they're going to replicate and get a similar pipe cleaner and join it to that first one. So they immediately see, oh, this is how replication occurs. It's now made a copy of itself. And then you can actually link these onto the spindle and put them in the middle so that you've got that metaphase stage. And if they line up in the way that they do, obviously, at metaphase, you can show how each individual chromosome lies as its replicated self on the middle of that spindle, and then show them separating and going one to each end. So what you end up with are 4 chromosomes at each end, and they're identical to each other and to the original cell. But I find that using an actual model spindle actually really useful. And these are the key points that they've actually got to learn. So again, before they worry about the names of stages, and the new syllabus actually asks them to know the names of the stages, whereas before they just had to know the sequence, but they need to know prophase, metaphase, anaphase, and telophase now. Which is quite a bit of extra learning, I think, and can be quite tricky. But let's forget those terms until the very end. My trick for mitosis, so they can remember it, is different from meiosis, is the idea that mitosis is making identical to. Meiosis is making eggs and, of course, sperm. So let's simply look at the key points about mitosis and meiosis. Let's list them there, and we've got the key points I've put on here, which I don't need really to tell you about, but these are the key points that a student needs to learn. How many cells are made? Why are they only 2 in mitosis? Why do we have 4 in meiosis? And the idea, of course, linking in that because you've got the extra stages, it allows for the crossing over processes and so on. So they get recombinations in meiosis, which they don't get in mitosis. And once they've got a reasonable idea of what's going on, then a homework exercise like this one can actually allow them to find for themselves the names of the stages, and then to make notes underneath each stage about what's actually happening. And you can see it goes on to say, well, what happens after T? Well, that's actually when the cell itself divides. So far, we've been talking about the nucleus dividing, but at the end of it all, you've got two separate cells because the cell membranes pass across and separate them into two. And then on this particular exercise, it suggested that they do the same for meiosis, and that's going to take a lot more thinking. So it will depend on your particular students how much help they'll need to actually do that flow chart. And the optional activity given in the textbook is what we've just been talking about, using pipe cleaners to make models and to actually sort out what's going on. And another tricky topic, again, because there are probably lots of names that are unfamiliar, is the circulation. So in the textbook, and I suggest you use simple diagrams to start, we've started with this very simple diagram. Very few names, just what type of blood have you got in the various tubes that are showing, and the heart itself. So a discussion perhaps to start with, without any names of the blood vessels, what's going on here? And if you've got a big enough diagram, you can put your finger on the diagram and trace round where it goes from one part to the other, and get them to obviously appreciate that there's this nice figure of 8 double circulation, and that if you start at the head, you've got to go to the heart first before you go somewhere else in the body. Now, so far, of course, we haven't got any of the main organs in, which is where it starts to look complicated to them. So if we think about, okay, the blood goes from the heart all around the body, goes to every part of the body, and then it's got to come back to the heart, and so on. There's a nice YouTube video, it's called Blood Flow Through the Heart in 2 minutes. But you need to check if that's available, and look for the most suitable video for your group. If the book suggests this is the search term, but actually they can waste a lot of time trying to find the most suitable. So it was very helpful if you can actually do it for them. And I would suggest that for many of them, videos designed for younger students are actually a better introduction than going straight to the more detailed ones. I found the Children's Hospital Colorado video, and I think that would be easy for you to find too. And it's actually called What is the Lymphatic System, which is something, again, that people find very abstract because they're used to blood, but they really don't know what this lymphatic system is. But the first part, there are 2 parts to this. The first part is the anatomy and the physiology. The second one is probably less suitable because it actually then deals with disorders of the lymphatic system and so on, which children who are suffering illnesses might want to look at to understand their particular condition. But the animations are very good in this one. They show the blood and the lymph circulating. They look like little Lego trains going around in the blood vessels, but actually they're quite engaging. And they also deal with the functions of the lymph. And although it's 6 minutes, I found it was 6 minutes actually very well spent. Again, we can use our bodies. And if a student doesn't know much about arteries and veins and so on, use the body in this way. This is a classic example of demonstrating venous blood flow and sometimes being able to actually show where the valves are. It's quite difficult to find a good demonstration of this actually online. I found one using the search term demonstration of venous blood flow. You may well have access to better ones. Maybe there are some links in the syllabus to various videos and so on, and resources. So you may well have a better one. But look, again, when they go into an exam, if they can't remember what's going on with arteries and veins, they've actually got them to look at, and maybe to prompt them to think about what they were doing in class and what it involved. And actually, why did we press veins? Oh, well, it didn't go backwards because there were valves in there. And so they're starting to think in the exam of actually what went on in the lesson by, again, looking at their own bodies, maybe pressing a vein and trying to remember what the actual example was in the lesson, what the demonstration was in the lesson. Once they've got some idea of arteries and veins and capillaries, they can use the more complex diagrams. And the one on the right is actually there, obviously, to complement the one on the left, so that you can see the main body structures, the main organs, know where they are. And then you can see how the blood flow and the circulation links to that particular diagram. But now it looks very complicated. We've got lots of names in there. How on earth am I going to learn all these names? Well, learn the key ones. And one of the things that you can do is actually pretend to be a sat-nav yourself, or get one of the class to say, right, I've got to give you instructions of how to get from the head and how to get to the liver using sat-nav instructions. So everyone has a diagram in front of them. They put their finger on the head, and then they follow the sat-nav instructions and say, right, okay, from the head, we need to enter the jugular vein, and that's going to take us down to the next junction, and the next junction, we enter the superior vena cava, and so on. If students do it, you have to obviously monitor that they're going in the right direction, but they can work in groups and talk to each other, and one person can be the sat-nav, and then they can change who does that job, and everybody tries to follow the pattern around the body. So let's start first of all at the head, get to the liver. What about going from the kidneys back to the lungs, and that sort of thing? And I think that can be quite engaging for many youngsters. Enzymes is another one that people are concerned about, partly because the time constraints and the constraints of having no lab facilities, or maybe the apparatus, can be quite a problem for HSB when the school perhaps has to manage biology as well, and they have access to the labs. So you can start obviously with enzymes with the bread experiment, and find out what type of chemical actually changes the bread, and start to discuss the idea that we need these chemicals called enzymes to change things. And this particular activity is just a starter for 10, if you like, so that you've got what happens to the starch in my mouth if they chew a piece of bread or a cracker. You may have to worry and check that no one is sensitive to any of the ingredients. But if they bring their own that they normally would eat, so if it's gluten-free, for example, you can still do this little experiment, or they can do it at home. And it encourages them to think about what's happening and start the work on enzymes. And as with the kidney and all the other topics, actually, start with basic terminology, develop understanding, and then start to introduce the new vocabulary. So the obvious questions there: the bread is changed, what does it change into? What type of chemical changes it? And at this stage, we don't even need to know amylase, but if you feel that they can cope with that, okay, that's one of the few enzyme names they need to learn. and develop what enzymes have to do before you bring in the new words again. And I suggested that if you haven't got time to do the experiments in class, you can either use an online ready prepared video, but of course it would be nice if you could actually produce your own videos, working perhaps with your biology colleagues, and do your own demonstration. And you could stop and start that so that you can talk about the parts of the experiment, talk about what's going on. Okay, so that's just a way of perhaps engaging them with enzymes to start with. The activity can be online. You can use your textbook. The textbook again gives them the answers, the results, and so they've got a visual idea of the experiment. Very often students will say to you, why am I learning this? You know, what's the point of enzymes? And we've got in the real world a whole list here of uses of enzymes. Some of them actually appear in the specification, the syllabus, but actually many of them are just for general interest. And throughout the book, there are examples of these real world topics that actually put their subject into context. For genetics, very quickly, I've suggested one of the things that they need is lots and lots of practice. And one of the problems we have is that we're not always sure whether something is dominant or recessive. And in fact, if we don't check and they come back to you and say, look, I read this and it says that's not true. So, for example, with tongue rolling, we always used to say that tongue rolling was dominant and the inability to roll the tongue was recessive. But actually, we've learned now that people can learn to roll their tongue, and it's sort of the ability to learn, and that gets more complicated. So if we use fictional characters, we can make up any feature that we like. I've just given the example of Star Trek. Dr. Spock has big pointed ears. He has a child with a human who has small ears. Big ears are dominant. So find out how many of their children will have big ears and small ears. Once learners realise there's a limited number of crosses, then actually they seem to enjoy this particular topic. For diseases, concentrate on what's common. There are many cancers mentioned in the syllabus, but actually we need to understand what a cancer is and how it can be spread. I'm rushing you through this because I can see that we're getting rather short of time, but you can see from this particular diagram that everything you need to know about cancer is actually there, that there are stages, that it can spread, and how it spreads, goes into the bloodstream, for example. And then you can worry about, well, okay, there are several reproductive cancers mentioned in the syllabus, but basically the way the cancer works is the same. So how do we incorporate blended learning into HSB? Well, the first thing you need to do is review the new syllabus. Some of the topics have been removed, others have been added. It's full of content, and that requires learning unfamiliar vocabulary. So which areas of that might be delivered using appropriate online resources? Which need face-to-face teaching to allow for discussion? Which topics could be enhanced by use of experiments, flashcards, quizzes, use of models, class challenges, group work, student presentations, outside speakers, demonstrations and field work and visits, such as going to a water treatment plant. The new textbook has actually attempted to incorporate blended learning in many of the activities where they're appropriate, has given suggestions of how to find online facilities so that you can actually use the digital component of blended learning. And the other thing I would suggest is that you create a scheme of work which has columns for all those appropriate activities. The more detailed that scheme of work is, I think, the more useful you will find it. So here are a few final thoughts. before perhaps we have a few questions. Be very careful when using older editions of the textbook. That applies to our textbook and other textbooks that you may well be using. Check that the content is still relevant to the new syllabus. As I said, things have been added, some things have been removed. And if you just go through the textbook in order as it's written, you may find that actually you're teaching some things that are not relevant. Detailed planning will save you time later. I think that's really important. That's what we mean by planning a scheme of work. And the more detailed the scheme of work, the more confident you'll feel. The experienced teachers in your school will be able to help those who've not taught the subject before. And if they already have a scheme of work with lesson plans, which includes the sort of amount of time you might expect to take on a topic, it can actually make you feel much more confident that you're going to cover the whole of the syllabus. The textbook, it actually offers more than something to read. It uses ideas from use ideas, I would say, for these suggested activities. You don't have to do everything that's in the textbook that we've suggested, but it might prompt you to think of something that will work really well in your school and with your particular learners. As I said before, it's always worth checking online materials are actually appropriate and suitable for this level. Can I suggest, we've got lots of people online today, that you share ideas with people right across the region. Maybe you could, I use WhatsApp, but you probably have your own platforms for sharing ideas. But maybe if you could share ideas with each other, you can also give each other confidence. You can say, well, I found this wonderful video. I think you'd find it useful. And then somebody else will say, how about trying this practical? I've got this quiz. Would you like to share that? And I really think that working with other colleagues is a wonderful way to actually progress in a new syllabus. As you can see, my final point is actually what the textbook offers in support. And it offers support for the CXC examination. It includes the in-text questions, which you're familiar with, but not the smaller green questions, which we used to have. We've combined all those together to one lot of in-text questions, and they're actually graded as well. And you can tell how the more difficult ones by different colours. We've got multiple choice questions, 16 per chapter, and they're trying to match the ones that you're likely to see in the examination, the style of them. And then the examination style questions, the paper 2 style questions, are there, the 15 markers that you're used to for the examination. So they've been redesigned so that you've got very similar sorts of questions, and all the answers are online. Teachers asked us not to put them in the book because they said their students cheated. So we've put them online, and you can choose whether or not they have access to it, or you give them the answers. And the other big support the textbook is offering now is for the school-based assessment. This is brand new for everybody, and we've got project skills and project topics. In other words, suggesting the sorts of things that might be useful for the school-based assessment. And there's a whole chapter on how to do the school-based assessment with examples of both good and not so good practice. Well, that's a very rapid hour, but I hope that some of you have found some useful ideas there. These are just suggestions, obviously, but it's how I've managed to combat some of the more difficult topics that my students have been confronted with. I hope you've enjoyed it today, and we'll welcome any suggestions for future topics. We've got a few minutes if there are any questions, Rainer. I'm reading some of the comments that are coming up there, and I'm noticing that quite a few people have missed perhaps the first slide of the new book. The new book is due to be published, and it's called Human Social Biology for CSEC, the 7th edition, and you really will need the 7th edition, I think, for the new syllabus, due to, as I said, to the changes not only in the content but also in the school-based assessment.
Perfect. Thank you so much, Neva. Sorry about that. Sometimes I have trouble with my microphone and modern technology.
I'm glad you're there.
I'm definitely here. I just wanted to say before we finish, just to say thank you so much to Neva. This is her first webinar that she's delivering for us in this format. So she's done a fantastic job for us, and also highlighted some key elements of our new HSV title. Neva will be delivering a session next week on Thursday, 25th of February, focusing on the key features of the new title. And just to let you know also that there are going to be interactivities included in the new title. We'll provide further information next week on this, so please do join us. You will receive further information as a follow-up email as well. And if there are any questions, feel free to let us know. But I think some of the questions have been answered already in terms of when this will be published, which is at the end of the month. And also information on how to contact us is on the following slide. So do contact us on our email address, and do check out our website for upcoming webinars, and also do join us next week for Neva's second webinar. So just to say thank you so much once again to Neva, and also to our attendees for joining, and we look forward to seeing you in an upcoming session soon. Take care for now. Bye.